The next subtopic that we are going to discuss under genetics is actually the process of meiosis. So, since this is being learned by both SL and HL students, it is better to to explain in a more friendlier way. It doesn't need much depth comparing to how HL students are going to learn them later on in the next chapter. So, in the case of meiosis, of course, okay, we have to explain the significance of the process of meiosis. Of course, meiosis is being used for the cell division or the production of gametes or sex cells. And as we all know that sex cells, they contain they contain different sets of chromosomes. They contain haploid chromosomes, haploid cells, because they only have the half amount of the total amount of chromosomes. So if we have 46, okay, 46 chromosomes, and therefore each sex cell should only have 23. It's because if the sperm cell and if the sperm cell is going to have 23 chromosomes, and the ovum is going to have 23 chromosomes as well. When they fuse together in the process of fertilization, okay, fertilization is going to restore the diploid amount of chromosomes later on. So it's not logical that the sperm cell will have 46 and the ovum will have 46 as well, because whenever they fuse together, it will have a different number of chromosomes in the end. So that's the significance of meiosis. And meiosis, apart from that, it also provides us variation. Okay, not unlike mitosis, the parent cell and the daughter cell are exactly the same. Okay, so in the case of meiosis, since whenever the homologous chromosomes line up together, there is an exchange of genetic information between homologous chromosomes, which give us more variation in the characteristics or in the traits that is going to be given to each and every cell. Okay. So next, okay, the next part that is going to be discussed in meiosis is we have to discuss what a homologous chromosome is. Okay, it's going to be helpful if we're going to teach them using like a different colored markers, because it represents, for example, if we're going to draw like, okay, assuming that this chromosome is coming from one parent, okay, and this chromosome is coming from another parent, okay, and assuming that they are of the same length, same size, and the same position of the centromere, then we can say that whenever this homologous chromosomes cross together, they still cross, or they still exchange same genes. Okay, so homologous chromosomes, they are structurally similar chromosomes. Okay, that contain the same set of genes. May or may not contain the same set of genes, but they're coming from different parents. So we have to reiterate to students as well that the reason why siblings, or for example, all offsprings in a family doesn't really look exactly the same as their parents, it's because whenever homologous chromosomes line up together in the process of meiosis, then they get different, each and every uh, sperm cells and ovum, they get the different set of genes. So whenever they cross together and they're being used in fertilization, they get different sets of genes. Okay, so the next one. Okay, of course, it is very important to discuss the process. Okay, but before that, we have to make an outline of what is going to happen in meiosis. Since meiosis is subdivided into two sub-processes, okay, so we should make an outline of meiosis 1 and meiosis 2. Okay, so we can also tell to our students that in mitosis, there is no occurrence of homologous chromosomes, whereas for meiosis, there are homologous chromosomes. So, okay, so homologous chromosomes are not used in mitosis. So, okay, we should point out that meiosis 1, okay, the goal of meiosis 1 is to separate homologous chromosomes. And in the end of the process, you are able to produce 2 daughter cells. Okay, and meiosis 2, this is where it separates the sister chromatids. Wherein it produces 4 daughter cells in the end. Okay. So, right after pointing out the outline of what the process is, then okay, we have to go to the main events. Okay. Okay, usually, the process of meiosis and mitosis, they have their similarities, but we have to point out their differences as well. So, in the case of meiosis, we have to point out that prophase 1 is it's a very long process. It is a very intricate process as well, because in the case of meiosis 1 or prophase 1, we have the crossing over. Okay, in the case of crossing over, this is an event of an exchange of genetic information between 2 homologous chromosomes. So this phenomenon gives us more genetic variation in the chromosomes. Okay, so we have to indicate the main events that are happening in prophase 1, metaphase 1, and anaphase 1 and 2. Okay, so actually these are the special stages in meiosis that needs more attention, that gives the chromosomes more variation. Okay, so although there are other genetic laws that is actually encompassing this this part of meiosis, we do not need to explain them even further in this area, because they are just required in the HL part. Okay, so right after explaining the main events, okay, we can now explain that certain events in the anaphase, okay, also brings about mutation. Okay, so because of non-disjunction, okay, non-disjunction, this is the failure of chromosomes to separate equally. Okay, so which actually results to mutation. So in this type of mutation, we need to show a diagram, okay, to students that non-disjunction can happen in anaphase 1 and 2 specifically. Okay. So we can show to the students that in anaphase 1, instead of the two pairs of chromosomes separating equally in anaphase 1, one of the chromosomes failed to separate, which results to, okay, so for example, if I have the diagram here, okay, supposedly there should be two chromosomes that is going to be separated here. But because of non-disjunction, okay, one of the chromosomes failed to separate, and they became a part of one of the cells. Okay, they become part of one of the nucleus here. So later on, this cell is going to be rejected. It's going to degenerate because it didn't receive the same complete set of chromosomes. But this one, okay, because it contained at least two sets of chromosomes, so they will still survive, they will still be used, but there would be a mutation. So we can show to the students that because of non-disjunction, Down syndrome can occur. Okay, so in Down syndrome, we can show that on the 21st chromosome, instead of having just a pair of chromosomes, there are 3 chromosomes that are present in the 21st chromosome. Okay. So we can also show that non-disjunction can also be shown in karyotyping. So karyotyping is basically a process of taking a photo of the whole chromosomes that is not undergoing cell division, of course, because in the case of karyotyping, we are lining up, we are showing that the chromosomes are lined up together according to size, and then of course we can show that since they are occurring in pairs, we can see that the 21st chromosome there are three chromosomes present, which indicates that this individual is having Down syndrome. Okay, karyotyping is also being used to identify the gender of an individual because it also shows the length of the sex chromosome. Okay, so karyotyping can be acquired through the process of some genetic screening processes, such as chorionic villus sampling and amniotic sac sampling. So that will be all for meiosis.